Hi everyone, my name is Christina Canino. I'm making this video for the extra credit project in BC 351. In order to understand protein structure, we need to first look at the characteristics of a single amino acid. I've color-coded the three main aspects of an amino acid, the amino group shown here in pink, the variable R group shown here in red, and the carboxyl group shown here in blue. The carboxyl and the amino group are going to stay the same throughout each amino acid, but this R group can change. And the variation in R groups is going to dictate much of a protein structure based on how they interact with one another. Note that these groups are all attached to an alpha carbon here in the center. I've also drawn in the phi and C angles that are between the amino and carboxyl groups with the alpha carbon. And that'll become important when we look at peptide bonds. So we know that proteins are polymers of amino acids, so it's valuable to find out how these amino acids come together. We know that this happens through a condensation reaction between two amino acids where one molecule of water is taken out and one peptide bond is replaced between the carbon and the nitrogen. This peptide bond has a partial double bond, as I've shown here with the dotted line, and this carbon-oxygen bond is also a double bond. Sorry for that little scribble there in the middle. But basically, because this is a partial double bond, peptide bonds are planar and restricted rotationally. And that's where the psi and phi angles come into play, because that's going to be how proteins move about within their structure. I have this larger extended model drawn here, still with the amino group and the carboxyl group at the beginning and end, with 4 different R groups all attached to alpha carbons. The green bonds are the peptide bonds, so remember those aren't going to move. And I've drawn blue and pink arrows denoting the phi and C angles in order to just kind of remind you that that's the part of the molecule that does have free rotation. The first secondary structure we're going to look at is an alpha helix. I understand this picture is probably a little confusing, but the important thing to note is that for an alpha helix, there's a hydrogen bond happening between the oxygen on the carbonyl group and the hydrogen on the amide group, and they happen between like 4 molecules in between. So I've numbered them here, so hopefully that helps. And as this bond forms, it's going to cause the protein to rotate about itself, and which way the oxygen to hydrogen bond forms is going to dictate whether it's right-handed or left-handed. And I've also noted the phi and C angles for an alpha helix there. And so those are pretty consistent throughout the alpha helix structure. It's also important to note that certain R groups do not work well at all with alpha helices, while others are very beneficial for alpha helices, and I've noted those there. When we look at beta sheets, that's the next type of secondary structure, also dictated by hydrogen bonds. How beta sheets form are between different strands of, or rather, I guess the same strand of protein, but running one along the other. And there's going to be anti-parallel, which we're looking at first. And so how that works is the hydrogen bonds are parallel to one another. They line up very well. And you can see that the 2 strands are going in opposite directions, and that's characteristic of an anti-parallel beta sheet. And if we look at a parallel beta sheet, there's still hydrogen bonding, but because the strands are going in the same direction, the hydrogen bonds are at a little bit more of an angle, and this makes parallel structures less stable than anti-parallel structures. The last structure we'll look at is beta turns, and this is still a hydrogen bond, as all secondary structures are dictated by hydrogen bonds. This one in particular is between 4, again the number 4. So if we look at our first amino acid, second, third, and fourth, the hydrogen bond exists between the first oxygen on the first amino acid and the amine hydrogen on the last amino acid. And that's What dictates the beta turns?